Good morning. Today I will be taking on subcutaneous mycosis. I think all of you know what is the meaning of mycosis. Infections which are caused by fungi are referred to as mycosis. So we can classify the infections caused by fungi into four major types. The first one is superficial mycosis, then we have subcutaneous mycosis, systemic mycosis, and lastly what is known as the opportunistic mycosis. So since superficial mycosis is already completed, I will be continuing with the second type, that is a subcutaneous mycosis. The fungi which are known to cause subcutaneous mycosis, they are known to invade from the superficial layer and reach up to the subcutaneous layers. So primarily they cause infection of the subcutaneous tissue by formation of sinuses. To introduce to you what is subcutaneous mycosis, the classical lesions that are seen in subcutaneous mycosis are primarily of the granulomatous type of lesions which are seen in the subcutaneous tissues. The fungi which are known to cause these kind of infections, they are known to inhabit the soil, and the type of injury that they cause is very trivial traumatic injury. Through this the fungi enter into the superficial layer, then they start further invading and they reach up to the subcutaneous tissue and they set up the infection. So there are different types of subcutaneous mycosis which comprises of RHINOSPORIDIOSIS, mycetoma, SPOROTRICHOSIS, CHROMOBLASTOMYCOSIS and PHAEOHYPHOMYCOSIS. I will be dealing with each one in detail. First, coming to what is RHINOSPORIDIOSIS. RHINOSPORIDIOSIS is a chronic granulomatous disease characterized by formation of large friable polyps. You all know what is a polyp? It is a large mass. So the organism or the fungi which is known to cause RHINOSPORIDIOSIS is Rhinosporidium seaberi. It is a single organism. RHINOSPORIDIOSIS is distributed in regions of Sri Lanka and also in parts of India, that is mostly in the south of India, that is Tamil Nadu, Andhra Pradesh and Kerala. The source of this infection is found to be stagnant water. The mode of entry is through the entry of the fungal spores which are found in the stagnant water, and when the person enters into this stagnant water while either taking a bath or just for recreational activities, the fungal spores are inhaled. So this is a picture which is showing a type of polyp. You can see that it's arising from the nose. So this is referred to as the nasal polyp. In the second picture you can see that there is a polyp which is arising from the conjunctiva. It's referred to as the conjunctival polyp, which all these are the subcutaneous tissues. So and lastly you can see in the third picture a polyp which is arising from the pharyngeal mucosa. It is known as the pharyngeal polyp or pharyngeal rhinosporidiosис. Coming to how we make a diagnosis of rhinosporidiosис, just by looking at the lesion or the large polyp in the patient we can make a diagnosis, but we need to confirm it that it is caused by the fungus rhinosporidiosис to be very specific while giving the treatment. So the sample that we take is a FNAC of the polyp and we subject it for histopathological examination, particularly using the mucicarmin stain. On staining and looking at the slide under the microscope, we find this characteristic spherules. Okay, they are referred to as the spherules. So what is a spherule? It is a large sporangia, as you can see in the picture, which measures around 350 microns in size, and this large sporangia in turn contains small numerous endospores, which are again measuring around 60 microns, 50 to 60 microns in size. So the entire thing, the large sporangia containing the small endospores are referred to as the spherules, and it is very pathognomic of rhinosporidiosис. Culture till today we have not been able to cultivate or grow this Rhinosporidium seaberry. Okay, so there is no culture media which is available to grow this organism. That was about the lab diagnosis. Coming to the treatment, depending upon the region the polyp is present, they can do a radical surgery, that is removing of the polyp, and the patient is put on dapsone. Dapsone is also the drug that is given to cases of leprosy. So this is about rhinosporidiosis, which usually can come either as a 3 marker for you or rarely sometimes asked as a 5 marker. That is a spherule. Coming to the second type of infection, that is mycetoma. Mycetoma is also known as Madura mycosis or Madura foot. It was first described by a person named John Gill, wherein he identified this condition in Madurai. So therefore it is known as Madura foot. What is mycetoma? It is a slowly progressive, again it is a chronic granulomatous infection of the skin as well as a subcutaneous tissue, which presents classically as a triad of swelling of the affected region. There is multiple discharging sinuses, and the discharge contains granules. So this is a picture of a Madura foot. Okay, so the region, again it is more commonly found in the foot. So you can see that there is a swelling of the foot along with sinuses. So sinuses are those openings where you can see there is oozing of some discharge, and these discharge will contain granules. Okay. Based on the type of etiology, mycetoma can be of 2 types. The first one is mycetoma, then the second one is actinomycetoma. If the mycetoma is caused by fungus or fungi, then it is known as mycetoma. If it is caused by a bacteria, then it is referred to as actinomycetoma. Actinomycetes, that chapter has already been completed for you all when we did bacteriology. So coming to the different agents of mycetoma, the grains which are produced in this discharge, they can be of different colors. So based on that we can clinically identify whether it is caused by a fungus or whether it is caused by a bacteria. So we will see the organisms causing mycetoma. So mycetoma, the granules can be of 2 colors, either black or white granules. Again, we can narrow down the diagnosis again by knowing which organism is producing which color. Okay, so like for example, black granules are produced by these fungi. Black granules are produced by Madurella mycetomatis, Madurella griseae, Exophilia ginselmi, and the Curvularia species. Similarly, the fungi which produce white granules, the names of them are Pseudoleishmania boydii, Aspergillus nidulans, and Fusarium. These were the fungi which is known to cause mycetoma. Now, this is just a brushing up of what has already been done for you. Actinomycetoma, either they produce yellow granules or the red granules. Yellow granules are produced by Actinomadura madurae, Nocardia species, Streptomyces somaliensis, and red granules is produced by Actinomadura pelleteri. So just by knowing sometimes the color that is being produced in the granules, we can identify whether it is a fungus or whether it is the bacteria, because again the method of diagnosis becomes different. Coming to the pathogenesis of mycetoma, the source of infection is through the contaminated soil. So when a person is walking barefoot or using his hands in the contaminated soil, that's when the fungus enters. So the mode of entry, again it is through traumatic inoculation. The areas affected is a foot. Most commonly it is a foot, sometimes also the hands. So the mechanism is as the fungus or the bacteria enters, there is formation of the subcutaneous swelling, which slowly starts enlarging, and the enlarged mass there is formation of an abscess. First there is a single abscess, then it grows into multiple abscesses. These multiple abscesses will have openings which are referred to as the sinuses. So they burst open to form multiple discharging sinuses, and these sinuses are the ones which release the different colored granules. Coming to the clinical manifestations, again it is different in mycetoma and actinomycetoma. Just by sometimes just taking the history or just by physical examination we can make a diagnosis. So what are the differences? In mycetoma, the tumor mass, it is usually single and it has a well defined margin. In contrast to that, in actinomycetoma, the tumor masses are multiple and the mass is very diffuse and ill defined. The sinuses, they appear late in case of mycetoma and relatively less in number, whereas it is the opposite in actinomycetoma. They appear early and they are more in number. The opening of the sinuses is flat and they are not flared up in case of mycetoma, whereas it is raised and inflamed and it is usually flared up in actinomycetoma. The discharge is serous or serosanguinous, whereas in actinomycetoma it is more of purulent discharge. The color of the granules, as I had already shown you in the previous table, it is black or white, and in case of actino it is red or yellow granules. The extension of the lesions, in case of mycetoma it is very less extensive. In contrast, actinomycetoma, it's more invasive, so it is more extensive and results in osteolytic lesions. The lab diagnosis, the samples that can be collected, first of all, is the grains from the discharge. Grains are nothing but the granules. Then if there is pus discharge, we can collect that. Sometimes from the tumor mass we can also do a biopsy and an FNAC. So methods of collecting the granules, the lesions should be thoroughly cleaned with antiseptics. The grains are collected by pressing the sinus from the periphery onto a sterile gauze, or with the help of a loop they can be pulled out from the sinus. They should be examined for their gross appearance like the color, size, shape and texture. We subject it then for microscopy and culture. Now in the microscopy it again differs. If you feel it is a mycetoma, we put it for a KOH examination. Otherwise, if you feel it is a bacteria, we subject it for gram stain. And culture also varies. We use SDA for the fungi and the other bacterial media for different bacteria. Coming to the microscopy, in case of mycetoma, this microscopy that we do is a KOH mount. Wash the grains with sterile water, crush the grains between the slides and add 1 to 2 drops of KOH, and you look for the hyphae with chlamydospores under the microscope. Whereas on gram staining, if you feel it is a bacteria, depending upon the clinical history or the clinical examination or the color of the granules, you can decide. Okay, so we subject it for gram stain. And what we see on gram stain? We see the presence of gram positive hyphae. You can see them in the picture as well. Coming to the culture, if we are suspecting fungi, then the culture of choice or the culture media of choice is SDA with antibiotics. Why are we adding antibiotics? To inhibit the growth of the bacteria. We incubate it at 2 different temperatures, that is 25 degrees as well as 37 degrees for 6 to 8 weeks. So depending on the macroscopic, after the 6th to 8th week, we look for the macroscopic growth. From the macroscopic growth, again we do a microscopy, that is a tease mount, and we identify the different fungi which could have caused the mycetoma. The entire list, each of the fungi have different characteristic appearances on microscopy as well as macroscopy. So ultimately we give the diagnosis as one particular fungus. In case of actinomycetoma, the grains are washed with sterile saline and then we put it onto a culture media, that is brain heart infusion, agar, or the LG media can also be used. And then it is incubated at 37 degrees Celsius for 4 weeks. After that, same thing, we look at the macroscopic appearance of the culture and subject it for a gram stain, and depending on the type of bacteria which give different biochemical reactions, we make an identification of the bacteria. Coming to the treatment of mycetoma, you need to subject the patient both for chemotherapy as well as surgical intervention needs to be done. The chemotherapy that we give for mycetoma is itraconazole, which is given for a duration of 8 to 24 months. As we know that these fungal infections, they don't subside very quickly when compared to bacterial infections. So, the patient is put on antifungals for a very long duration. Surgery, again, it depends on the extent of the disease. If there is no bone involvement, you can just excise the mass. If there is a minimum bone involvement, you can just do a debridement or a bone curettage, and then followed by a split skin grafting. If it is too extensive, then we go in for amputation. In case of actinomycetoma, we put the patient on antibiotics. So the combination that is given is amikacin with cotrimoxazole, again for a long duration, that is for 9 months. This is about mycetoma. Both mycetoma and actinomycetoma commonly asked question for 5 marks. So when asked for 5 marks, you need to write both actino as well as mycetoma. Coming to the third condition, which is known as sporotrichosis. This is also known as the rose gardener's disease. Why? Because it is most commonly seen among the gardeners who are working in the rose gardens. Okay. So, it presents as a subcutaneous nodulo-ulcerative lesion. The causative agent for sporotrichosis is Sporotrix shankai, which is, I think you are familiar with this name because it is one of the dimorphic fungus. So what is a dimorphic fungus? It is a fungus which exhibits dual morphology at 2 different temperatures. It appears as a yeast at 37 degrees Celsius or the body temperature, or it appears as a mold, okay, the one with the aerial growth at 25 to 27 degrees Celsius, that is the room temperature. Sporotrichosis is common in the Himalayan hilly regions, usually along the belt of Himachal Pradesh to Assam. So what is the pathogenesis? The source is here the decaying wood or the bark of the tree or the leaves. The mode of entry is commonly through the prick. Okay, it could be a thorn prick, like for example, why it's called as a rose gardener's disease. The thorns of the rose plant can sometimes have the spores of this fungus. So it is following a thorn prick or following a splinter injury. So it is also common among the carpenters. So the mechanism is the spores of this fungus, they get introduced into the skin following this trivial trauma. The enzymes are secreted by the fungus. It is the proteinases. They cause local invasion and then they cause local destruction in the subcutaneous tissue. Then they start traveling or spread along the lymphatics of the affected region. Therefore, you can see that the nodules or the ulcer is found along the line of the lymphatics. This is a picture where you can see the nodulo-ulcerative lesions on the hands. Coming to the lab diagnosis, the sample that is collected is the pus aspirate from the nodules. We subject it for a KOH mount wherein we see the elongated yeast cells. They are referred to as the cigar shaped yeast cells. Again, we can see only this kind of morphology because at 37 degrees Celsius, sporotrichosis appears in the yeast form. And since the sample is taken from our body, it's at body temperature, that you can see only the yeast forms. When we sometimes also subject it for the histopath examination using the H&E stain, we see the classical asteroid bodies. Okay, so what are these asteroid bodies? You can see in the picture on the right hand side, there is a central yeast cell which is surrounded by the antigen antibody complexes. Okay. Coming to the culture. Culture is always the gold standard or the confirmatory test. So when we collect the sample, we put it onto the SDA media and we put it into 2 tubes. One tube is incubated at 37 degrees Celsius and the other one is kept at room temperature. So we will see what are the growth patterns. At 25 degrees Celsius, macroscopically, you can see that there is a cottony growth which is black in color. And when we do a microscopy from this culture, we will see mycelial form with slender, delicate hyphae with projecting sterigmata. Okay. The thin long ones that you see is the delicate hyphae. From these delicate hyphae are arising these sterigmata, and these sterigmata at their ends they bear these conidia which are arranged in a flower like pattern. This is very classical of SPOROTRICHOSIS. This is at 25 degrees Celsius. Coming to what is the forms that we see at 37 degrees Celsius, macroscopically you will see creamy moist colonies grown on SDA, and when we do a microscopy from this, we will see the cigar shaped E cells. Okay, so since it's a dimorphic fungus, you will see 2 different morphologies being exhibited at 2 different temperatures. The treatment for SPOROTRICHOSIS, we use what is called a saturated solution of potassium iodide. It is in the liquid form which has to be taken orally 5 drops 3 times in a day for 12 weeks, and it has to be consumed with milk or juice to avoid any kind of gastrointestinal side effects. The other drug that we give them is the itraconazole, which is an antifungal, again for a duration of 2 to 4 weeks. Coming to the next subcutaneous mycosis, that is CHROMOBLASTOMYCOSIS. This is again a slow growing subcutaneous lesion caused by a group of demataceous or phaeoid fungi. That means these fungi, they themselves are darkly pigmented. So they are nothing but the color producing fungus. So the agents which cause chromo means color producing. Okay. So the agents which are causing chromoblastomycosis are Fonseca pedrosi, Fiolophora varicosa, Cladosporium caryonae, Rhinocladiella aquaspersa. So the lesions which are seen in chromoblastomycosis, they are of varied type. Either they can be varicose type, they can produce ulcers, the ulcerative type, or they can just form nodules which are known as the nodular type. The lab diagnosis in this case, again we take a biopsy from the lesion, subjected for KOH mount, and we see the classical characteristic sclerotic bodies. So what are these sclerotic bodies? They are round cells with the presence of internal septate. When we subject the sample for culture, we see that the culture, they become darkly pigmented because as such the fungi produce color. So they become darkly pigmented and they have a woolly growth. The treatment for this is surgical removal of the mass, and you can also put the patient on itraconazole. All this, what was shown in the previous slide, it is just a recapitulation. The varieties of lesions, the varicose type, wherein you will see so many rugae which is on the lesion. If there is an ulcer, it is the ulcerative type, and then if there is formation of nodules, it is the nodular type. When we take a biopsy from these lesions and subject it for the KOH mount, you will see the classical sclerotic bodies, which are also referred to as the copper penny bodies. Okay, you can see that the one cell itself is divided into 3 sections by the presence of internal septate, and then the growth on the culture media is darkly pigmented and it is woolly in nature. Coming to the last one, that is PHAEOHYPHOMYCOSIS. These are again slow growing subcutaneous lesions which are caused by again a group of demataceous or phaeoid fungi other than that described in CHROMOBLASTOMYCOSIS. As the name itself tells you, pheo means phaeoid, that means pigment producing fungus. So the agents which are known to cause PHAEOHYPHOMYCOSIS are Alternaria species, Bipolaris species, Curvularia species and Exophilia ginselni. Okay. This can rarely be asked for 3 marks. Okay, whereas the rest of them, they can be asked either for 3 marks or for 5 marks. So this is where we complete subcutaneous mycosis. Thank you.